Hi everyone, this is Mrs. Ortega. I hope you're doing well and are excited that this is our last lab session that we'll have before the practical. So we are almost there at the end of the semester. It's coming around quickly. So with that said, usually this last class we would go through our material. You guys would do your assignments and then you would have a completion grade. And so that's why for our online version of this class, I'm going to do this video. You guys will watch the video and then answer the question that will be given later on in this video. So it's pretty much it's very similar to what we could do in class, and that hopefully you'll receive a little bit better knowledge and information after having watched the video today. And so today we'll take a look at chapter 27, which is basic animal tissues. This is a topic we did cover in lecture already, so hopefully you are remembering the material, and now we will just take a look at how we apply this in a laboratory setting. But a little bit of review here. Tissues. Tissues are defined as collections of specialized cells and cell products that perform specific functions. And so we'll look at tissues and how they have different functions with their different structures and how their structure has always correlated well with their function. And the study of tissues is called histology. There are a lot of histologists out there that will study tissues in terms of medical field. There are medical technicians that will identify tissue samples and can study them and identify, if it's cancerous or benign or what type of maybe illness or disease that tissue sample has based on histology. So it's a very important, very valuable subject as we're studying our own tissues, our own skin, our own cells. So tissues combine to form organs, like the heart and liver are made up of many different tissue types. And so most organs actually have all four types of tissue. And so those four types are epithelial, connective, muscle, and nervous tissue. And so if you see here, this is just a diagram that kind of outlines how this forms. Cells themselves secrete and regulate an extracellular material or matrix and other fluids that surround the cells. This combines with the cells to form tissues. Tissues perform specific functions, as we see. Epithelial tissue covering exposed surfaces, connective tissue providing support, filling spaces, muscle tissue producing movement, nervous tissue, electrical impulses with information. All of these things are very, very important. Tissues, these combine to form organs with many functions that interact in organ systems. And so let's take a look at the first tissue type, epithelial tissue. So they have many functions, that some of which are listed here. You can see it provides physical protection. So our skin provides protection from the outside environment, as well as our internal protection. Our organs are designed purposefully to help each other and then also to protect each other. The stomach acid is not going to accidentally get into the small intestine or other organs with a very specific purpose. They have very specific structures that allow for physical protection from internal and external. Controlling permeability. So this is very, very important for gas exchange, as we saw in lecture. So controlling the movement of carbon dioxide and oxygen out and into cells is very, very important, and is done by epithelial tissue. Providing sensation. In terms of external epithelial tissue, our skin, we can sense some slight movements and detections very easily. So our skin is actually highly innervated, which means have nervous tissue, and so that provides very acute detection, which is also known as sensation, one of many things. They can also produce specialized secretion. So epithelial tissue actually has a second kind of subtype, which is gland. So glands are structures that produce fluid secretions. An example here is our goblet cell secretes mucin that combines with water to form mucus. And so that mucus is needed by many internal organs like the stomach to protect the lining of that tissue from the stomach acid. In other areas, goblet cells are needed as well. So here we see also epithelia as a singular form, epithelium is plural. These are defined, this is defined as layers of cells covering the internal or external surface. And that can be exposed surface and internal passageways. And so for epithelial tissue, the main takeaways here is that it's classified based on number of layers and shape. And so here we clearly show the number of layers. If it's one layer, it's simple. If it's two or more, it's stratified. So one layer or two or more, it's very usually pretty easy to detect if it's one layer versus if it's more than two. And then for shape, we've looked at shape before. There are three main shapes you'll want to know. Squamous is the flat tile-like shape. You can see in this image it has a kind of hexagonal outline with on the side view you can see the nucleus kind of popping out. So it kind of looks like a fried egg to me, or some people think tile or scale. It's very, very flat. And so this we'll see is very important for gas exchange. Cuboidal is the second shape. You can see it has the nucleus in the center with about equal amount of cytoplasm or filling on each side, and it's in a square shape, and so that makes it cuboidal epithelia. Then lastly, columnar. That's going to have a more rectangular shape, and end is going to have the nucleus towards it, and then the other end is going to be more cytoplasm, which you see there. And so looking at the epithelia here, starting out with simple squamous, for lab, you will want to be able to identify these cell types from images. And so here, and not limited to the images you see, but if I pick a different image, it will be something that will be clearly defined, will be a nice clear image that you'll be able to identify based on what you've learned here. And so looking at simple squamous, that's one layer of the flat-like cell. The side view here you can see in real images can be very hard to see because it's so flat. You're looking for really the cell shape and then the nucleus, which is that nice dark purple circle within the whitish lavender purple circle. And so that is going to be simple squamous, one layer of the flat cell tissue here. And so this one, from the side view, you can see it's connected to a basement membrane underneath. That's going to be tying it in with other tissues, other organs. And the outside is the free surface on top. From a bird's eye view, it's a little bit better, a little bit easier to identify. In that here you can see that these cells are connected close together. They have that shape, the hexagonal-like shape. It can be a little bit difficult to determine if this was squamous or cuboidal from this view, but knowing that it is from a bird's eye view, you can imagine just a flat tile-like cells all next to each other. That's what you're seeing here. And so it can be a little bit difficult in terms of which view, and that's why I will label those views for you to help prevent confusion. All right, so taking a look at cuboidal here. I'm sorry, I forgot one thing here on simple squamous before I move on. These cells are needed for gas exchange, so they're found in the lungs, in the heart, and other regions where blood vessels, where you need a very thin layer of cells to kind of define a line, so to speak, but you also need them to be able to move things quickly through them. And so moving carbon dioxide and oxygen into and out of the blood inside the lungs is important, as well as to the blood vessels to the rest of the body. And so simple squamous will be found in the lungs, and the blood vessels like the capillaries will have these simple squamous cells lining the capillaries and other regions. So moving on to cuboidal here, you can see from a side view and the bird's eye view, these are much more square, and that those nuclei are usually pretty well close to the center and take up a lot of space. They are usually found in glands and ducts. So that's where you want to look for them, are glands and ducts. And so this one here, you can see from the side view, this is a duct, as it is a passageway leading to an area where fluid can be secreted to, or where fluid is being made. Whereas this one here, depending on the sample tissue, it could be the gland itself that's been cut, or a duct. And so it's a nice circular shape, so we can classify this one as a gland. If you can imagine cells being on the end of this right here, then it's producing a fluid, and that fluid will be leaving through a duct. And so that I could classify as a gland. But the circular shape and the duct shape, the passageway shape here, is what you look for cuboidal cells. So these will be found in different regions. So cuboidal cells can be one layer or two layers. So for one layer, so a simple cuboidal cell, you can find them in the kidney tubules and the thyroid gland. For stratified cuboidal, you can find them, they're very rare, but you can find them in sweat glands and larger ducts of mammary glands. So moving right along now to the columnar cells. So as you can see, most of the images will come from a side view, as the bird's eye view is not beneficial in this case, to see that these are rectangular, they're long. Some of their nuclei can be very, very large, as you see on this image on the left. But some of them can be smaller and be more closely to the bottom of the cell, which is shown here in the right. And so this bottom part is actually given the name of basement membrane or basal surface. And so it's kind of depending on where you're looking, basal surface or basement membrane. And so that's just to help you identify that area there. And so for columnar, most of them will be simple. These are going to be found, so one layer, they're going to be found in the stomach, in the small intestine, the gallbladder, uterine tubes, and kidney collecting ducts. So they could be found in many areas. They usually will have microvilli Remember those tiny hair-like projections on the cell are called microvilli. And so those are going to be usually pictured as well. I think they might see some here. Very, very hard, but this one here does not have that. All right, on to our pseudostratified ciliated columnar cell. This is a mouthful. Let's break this down here. So pseudostratified means false layer. This one it makes it look like there's more than one layer, but it's actually just one layer of cells. So that's pseudostratified. Ciliated, that's your hair-like projection. So pseudostratified cells will come ciliated, and they will be in columnar shape. And so that's the rectangular shape. And so here you can see two images of pseudostratified ciliated columnar cells. But if you wanted to see like a diagram image of what pseudostratified really what it looks like in a diagram, it would be kind of like this, but probably a lot better, in that one nucleus of a cell is going to be by the basement membrane on the bottom here, and then one of them is going to be towards the top, and then the next one is going to be, towards the bottom or in the middle, and it just keeps going with different regions of where the nucleus is. And so that makes it look like there are many layers, even though there's not. And so that's pseudostratified. It looks like many layers, but there's just one. And so it can be harder to see in these real life images here, but you want to look for the basement membrane. You want to look to see if you can find any separation of cells. So if there was a clear ending of the cell right there or right here, you would know that it's actually a stratified, something other than pseudostratified, but you don't see that. And so that is how you identify pseudostratified. You look for the nuclei being in different regions here, and so that's a key indicator. When it looks like a disorganized nest, it can be pseudostratified. It could be something else, but we'll keep it to some simpler things here. So pseudostratified can be found in the nasal cavity. It could be found in the trachea and bronchi and parts of the male reproductive tract. And so they're here for protection, secretion, and moving of mucus. So you can see some goblet cells more clearly in this right image. Right here is a goblet cell. And right there as well. So that's forming mucin that will then be released, combined with water, will make mucus. All right, moving on to our stratified squamous epithelium. So this can be found in two versions. The first version is a keratinized version, which that word is right there for you, keratinized. This is a tough, water-resistant layer because keratin is a protein that's packed with filaments. This will be going to prevent water from leaving, as well as can be entering it as well. And so this is found on our skin. So we have here some stratified squamous epithelium example from human skin, and you can see there's the dermal layer, which is towards the more inner part of the skin. You have the epidermal layer on the top, and then you have the keratin layer of cells. So these cells are completely encased with keratin. They're usually dead at that part, and so they are functioning solely for protection from the outside environment. So there is a second type of stratified squamous cells. They are non-keratinized, and this is where they're going to be high areas with a lot of traffic and movement. And so regions of the mouth, pharynx, esophagus, anus, and vaginal regions will have stratified squamous epithelium for protection, as those cells can get shed easily and produce more quickly. And so areas of high abrasion will have stratified squamous cells. And so you can see this would be the epidermal layer here. And then this is, I'm not exactly sure where this example came from, but it is a nice example of a very highly impact stratified squamous cell layer. All right, so that wraps up our epithelia section. Now we'll move on to connective tissue. So connective tissue has a lot of different functions. This is the most diverse tissue type. As we'll see here in these next images, I'll point out what to really focus on. But in terms of overall ideas of function, this page is great in that you can see it functions and support. It has bone tissue, cartilage tissue, ligament, tendon, capsules encasing organs. So like the body cavities will have connective tissue, organ stroma, which is just the different connective tissue making up organs. That's all can be there for support. It's also a medium for exchange of metabolic waste, nutrients, as well as oxygen, as you find blood in here. So exchanging oxygen, carbon dioxide is done by blood, which is part of the connective tissue, which also functions in defense and protection. So blood cells and then the physical barrier that connective tissue provides will function in protection. And then storage of fat. So adipose tissues are types of connective tissue. And so the next two images here show you exactly how diverse connective tissue is. So it's made up of cells, which are highlighted in green here, and then also the matrix, which is that fluid we were talking about at the beginning. Fluid could be further divided. This matrix can be further divided into ground substance and protein fibers. So all these two images are very informative. They're not something I ask you to memorize, so don't memorize them, but do refer to them for your understanding of this section. So looking at cells here, cells are further divided based on whether or not they're fixed in one location, stationary, or if they're mobile. So if they're mobile, they're moving around the body. Those are blood cells. They can be further divided into red blood cells and white blood cells. So these are erythrocytes. You'll see this term in this PowerPoint, so you want to understand that erythrocyte is red blood cell. White blood cell is leukocyte. And so the red blood cells are the ones you're familiar with that transport oxygen and carbon dioxide. The white blood cells are going to be fighting invaders, foreign particles, bacteria, objects. Those are going to be functioning in the immune system. So they look different. You might have seen them before, but they're not the typical red blood cell shape that you're probably familiar with. Then looking at fixed cells, we have macrophages, which can be both mobile or fixed. It depends on the type of macrophage and where it's found. But they're fighting invaders by engulfing particles. Adipocytes, those are the fat cells. So key idea here, anything ending in site is going to be a cell. So adipocytes are fat cells. Then fibroblasts will make matrix, which you see here. For ground substance, you have mineralized matrix, which is bone. You have gelatin or syrupy matrix, which have the loose connective tissue, dense connective tissue, cartilage, and adipose tissue, which one we'll look at. Watery is the blood plasma found in our circulatory system. Then matrix is going to have protein fibers, and so these are some protein fibers: fibronectin, fibrillin, elastin, and collagen, lots of which you may be familiar with, are found in the matrix. So again, none of these diagrams here are things to memorize. Don't memorize them, but do use them to help your understanding of connective tissue and how diverse it is. And so in this section, we'll take a look at some examples of connective tissue, as we cannot look at them all. That would just be too much information. And so the first connective tissue that we'll look at is adipose tissue. So adipose tissue, you can see here, is responsible for storing fat in the form of lipids. And so lipids are going to be clear in appearance. And so here it looks like a big empty web, and that is what you're supposed to see. And so these things here you see this example here is what we have in lab, and it's not super helpful as you can't really see the adipose cells that well, as it's not stained very well. But if you were to look very closely, you can kind of see some clear outlines. Those are what you would be looking for there. But the better image is the one on the right here that's stained, so you can see the cytoplasm, you can see the plasma membrane, which is outlined in purple here. And then you can see where nuclei would be, which is kind of in the creases and the cracks on the ends of the cell. And so the cell is simply a big round circle with a plasma membrane holding it all together. And this is containing all the fat, all the lipids, and then the very tiny nucleus on the edge of the cell. That way it can perform all of life functions. And so here, that's what you're looking at. It's adipose tissue. You want to be able to recognize what this looks like. Then moving to connective tissue, the next example we'll see is hyaline cartilage. Hyaline cartilage is going to have cells that are suspended in a matrix, and so that's a very good example for connective tissue and how it's defined by cells are suspended in a matrix. And so that's what we're seeing here, is that these chondrocyte connective tissue cells are in a cavity called lacunae, or lacuna for singular. And so outside of that lacunae is a matrix. It's a suspension of watery or syrupy fluid. And so that's what you see here. Hyaline cartilage has cells, chondrocytes, in a covering or casing the lacunae, or in cavity lacunae, and then have the matrix everywhere else, which is that watery fluid. And so you can see that here in our lab image as well. It's just a little bit harder to see the matrix, as there are many, many cells in this hyaline cartilage area. Then looking at ground bone, so ground bone is going to be, there's actually two types of bone, spongy bone and ground bone. And so ground bone is the most dense, is going to form the hardest parts of the skeleton. And so it's made up of these structures that we see here, these whole circular units, like this one is called an osteon. It's just a units of bone. And then inside of that, the very center, there's a central canal that contains blood vessels. Obviously, these blood vessels are important for transport of nutrients, waste, as well as in the center of bone, it lies bone marrow, which is the manufacturing center of blood cells. And so transport through bone is very, very critical. And so these central canals play a huge role in that. Then looking at the osteon in terms of structure, it's made up of many rings, kind of like if you cut open an onion, it has very many layers. These rings are called lamellae, which is here. And so lamellae are rings of ground bone that are going to be connected to each other through lacunae, which are here at the bottom. Just like your hyaline cartilage, lacunae are going to be cavities that will host or house osteocytes. So these are ground bone cells, and they will be in these dark spot regions that you see here. And so this image is a little bit closer up. Hopefully you can identify it a little bit better and those features there. All right, and so next thing on connective tissue is blood. So here we have example of human blood and frog blood is a very different structure. So I like to look at ours and then a very distinct different one. So looking at human blood, you can see this is a very unmagnified image. So what you're looking at is a bunch of red blood cells. You know, the typical standard shape kind of looks like a donut with a whole middle filled in, right? That shape is our red blood cell, and you can see many, many, many of them here. And those are erythrocytes. The white blood cell functioning in defense immune system, that's a leukocyte, and that is in purple for the human blood. And so identifying which one's which is something you want to be able to do. That human blood, the erythrocytes are the red blood cells. These are functioning in transport, and so those are the red ones you see here. And then the leukocytes, those are white blood cells, are also functioning in immune system defense, and so those are purple here. Now looking at frog blood is very different. Their erythrocytes, or red blood cells, will actually keep their nucleus at maturity, which is something that ours do not do. And so they have a nice purple nucleus with a very clear cell, which is undyed here. And so you can see that you see these purple circles, and then around the purple circle you might see kind of like a pinkish circle, slightly darker circle, and that is your erythrocyte. That's a red blood cell for the frog. The leukocyte here is completely purple. So if you look at that, it's a little bit lighter in color and a much bigger circle. And so that is your white blood cell for the frog blood. All right. So now on to our third type, our muscle tissue. So muscle tissue consists of three subtypes: skeletal, smooth, and cardiac muscle tissue. And so these are pretty much similar to what we saw in the lecture, that we'll look at them based on how they appear. And so looking at something called striations is important. And so striations are alternating of light and dark bands. And this is due to the arrangement of proteins, actin and myosin, that will move, and these proteins will connect by hinging and connecting together, moving in different ways. They're going to move the muscle cells. And so muscle tissue responds to the nervous system, or neurotransmitters released by neurons. And they can also respond to the endocrine system with the hormones circulating in the blood. So they are responding to both types of signals. And so on this image here, we can see the type, the function, and the description. So in lecture, we'll focus on the function more or less, and in the lab, we focus on being able to identify, and so the description helps with that. And so looking at smooth muscle, you can see that these are spindle-shaped cells. So they're going to be kind of long, thin, and they're going to taper off at the ends, and then you have a long, skinny nucleus in the center. And so they have central nuclei, so nuclei in the center, no striations. They're going to be one color, no lines. And then they are very closely arranged, so very tightly packed to form dense sheets. And so this is helping in moving substances or objects along internal passageways. And this is under involuntary control. Now looking at cardiac muscle here, cardiac muscle is found in the heart, used to pump blood into the circulation. And so here they have the striated lines moving up and down that you can kind of see in this image. So different color lines. These are striations. And then they have uninucleate cells. So every cell will have a nucleus, one nucleus. And then they're going to have what we call intercalculated discs. These are special regions where special junctions where cells come together and communicate. And so you can see that kind of pretty well here, maybe here, here. And so that is, those are not striations, those are intercalculated discs. And so that's how you're going to be able to determine cardiac muscle from skeletal muscle, as they both have striations, more of which you can see here. These lines are more clearly defined for striations. Skeletal muscle is going to be much bigger in that they have many nuclei per cell, and their cells are much larger for obvious reasons, that they want to keep communication going quickly, and forming larger cells will be better for that here. And so their nuclei are flat, and they're usually on the end or the outermost part of the cell. So they're not be found in the center, they're going to be found on the side. And so that is going to be a key indicator there. And so they're going to have multinucleate cells as well, so many nuclei per cell, and they're going to be spaced out. And so they're going to be like regions of where they can send out proteins and send out other things to that area of the cell. And so this is obviously skeletal muscles for voluntary movement. Any voluntary control will be done by skeletal muscle. All right. So on to our last tissue type, nervous tissue. So nervous tissue is going to be coordinating body systems. It's going to be monitoring internal, external environment, and known for sending signals quickly in a matter of seconds or fraction of seconds. As we learned in lecture, there are two cell types: the neuron cells that are responsible for transmitting these signals, and then the glial cells, which support the neuron cells. And so for the neuron cell, we'll look at its different aspects. It's composed of a cell body, like any standard cell, but it's going to adapt to have the dendrites and the axons. The dendrites are going to carry impulses toward the neuron cell body, and the axons will carry impulses away or signals away from the body of the cell. And so they're defined by what impulse, where it's going. So if it's going to the neuron cell, it's the dendrite that's taking it there. If it's leaving the neuron cell, it's an axon that's taking it out. And so neuron cells are divided based on how this direction is done, or how the signal is received and sent out. For the one direction, so where the signal is received, then it's automatically sending out a signal. That is going to be the unipolar neuron. And actually, most of them are what we call pseudo-unipolar. And so it's a false one direction travel, and that the dendrites are receiving the signal here, then it's going down, and then off to the cell body is off to the side, where then they go down, get that signal, send out the new signal, and go to the axon. So overall, there's one direction that's happening there, but it's not a true unipolar neuron. The bipolar neuron is essentially two directions. So the dendrites receive the signal here, and then they send the signal out in a separate direction. And so technically, it looks very similar to the unipolar, but keep in mind, unipolar is kind of false unipolar. It's not a true one direction that we classify these directions as, but is what is found more often in the body. And then lastly here, multipolar is going to be receiving signals from many directions, and then can be sending it out to several directions, to several areas. And so that's what you see in that last one. So they're receiving from many directions, and then they're going to send out to different directions. And so here in our lab class, we have an example of the multipolar neuron. It is hard to identify in real life and in real samples, as it is integrated with other tissues. It's integrated with a lot of support tissues, the glial cells, which you can see here, and other just other cells in general. And so here you can see I've labeled the nucleus of a glial cell for support, and the nucleus of a neuron cell, and that this larger structure is a neuron, and that is supporting nerve glial cells are nearby it. And so that brings us to the end of this video, and that the end of our material. And so that brings me to your homework question. And so I wanted to kind of give you some background information about how I came to this question, and that normally I do a completion grade. And so instead of a completion grade, I want to kind of ask you a question as evidence that you did watch this video. And so I wanted to do a fun but yet science-related, topic-related question for you. And so I came up with this question, and the question is, if you were to shrink down to the size of a very, very, very small bacteria, which tissue would you like to explore, and why? And so lots of people are familiar with the Magic School Bus. There's an episode there where the teacher takes the students into, they shrink them down, go into the human body and explore. And so that kind of started this question: if you could Magic School Bus your way into one of these tissues, which one would you go to first, and why? And so if you answer that question, that is what will get you your completion grade for this assignment. Wanted to do something a little bit fun, but also something that got you thinking about this topic. So I hope you enjoy it. I hope you've enjoyed this lab class as much as you can, as now it's converted online. And I will be making announcements about assignments and other things like the practical coming up. So, this brings us to the end of the video. So, bye-bye for now.